So I've always wanted to be a scientist ever since I was a little boy. In fact, I've always wanted to study memory. And I think part of the reason I wanted to be a scientist is because my father was the scientist. And so I grew up with science in the family. And we used to have discussions. And when I was an adolescent, maybe they were more arguments. But one of the discussions that we had, I remember I was probably around 14. And the discussion was about which is it, if you're trying to discover how diseases work and to cure diseases, is it better to be studying the diseases themselves or to be studying the fundamental mechanisms of biology? And my father, who was a PhD, basic scientist, studying fundamental mechanisms of biology, he argued that that was the better approach. And that the diseases, figuring out diseases, would be something that comes later as a result of these fundamental discoveries. I argued, as a 14-year-old, I was thinking, my argument was, well, they're both hard, right? It's really hard to make fundamental discoveries, and it's really hard to understand diseases. So if they're both equally hard, why not just immediately study the diseases? And my father said, well, that's just not the way it works. So I thought that I won that argument. But as I went to medical school, did a residency in neurology, and then started studying basic mechanisms of neuroscience, I realized that, well, actually my father was correct. And it is, the way it works is that the fundamental discoveries reveal the basic mechanisms that then go awry in diseases. And that it's always been that that's the way in which diseases are being cured. It's the low-lying fruit, as they say, of a fundamental discovery. And the more fundamental the discovery is, the more fruit it bears. So part of the reason that my father was right, as I've discovered, is that one of the advantages of understanding fundamental mechanisms in biology, maybe in all of science, is that you can use beauty as a guide. So what do I mean by that? And why can't you necessarily do that when you're trying to figure out the causes of disease? So here's an example. And this was actually something my father had shown me in a paper, and this was when I was still a resident in neurology, and it led to the rest of my career. Because it struck me as an incredibly simple and beautiful way in which memories could be stored, which, as I said, I was always interested in understanding. So, as I mentioned in my talk, there was an enzyme called protein kinase C that had a catalytic domain shown here in green and a regulatory domain shown here in red that inhibits the catalytic domain. And normally, second messengers, which activate the enzyme transiently, just for a few seconds, will bind to the regulatory domain, cause a shape change that allows the enzyme now to be active. But this is a temporary phenomenon because these second messengers are metabolized in the cells, they're eliminated within seconds, and then the enzyme folds back up into this inactive state. And so the activation of this enzyme called protein kinase C is a reversible phenomenon. And that's important for signal transduction. it is like the keys on your keyboard. They all have springs underneath them, keeping them in the off state, so they could be used over and over again. So signal transduction mechanisms are important for transmitting information from the outside of the cell into the inside, but they're not information storage devices like the hard disk of a computer. But it was from the very first papers on PKC, they realized that there was a way to make this enzyme permanently active. It could be cleaved in half, releasing this catalytic domain, which was now released from its auto-inhibition from the regulatory domain. And so this catalytic domain, which was called PKM, was a way of now persistently activating the enzyme. And it acts as an information storage device, because this is an irreversible reaction. So I thought, when I saw this, that this was just beautiful. And so when I started my lab at Downstate, that was the first thing I did, was to see if there was any PKM in the cell. And what we found was that the whole situation seemed a lot more complicated. There were actually 9 different genes, and turned out only one of them, the zeta isoform, had this shorter PKM fragment, shown right here. So it was beauty that led to the hypothesis that there might be a simple mechanism for memory, but then the data were more complicated. And why was it that there was only one PKM form out of all of the 9 different genes of PKC? Well, the answer is actually itself even more beautiful. It's more complicated than the simple cleavage of PKC into PKM, but it's more beautiful in the deep biological sense of it has much more explanatory power. So this turns out to be the mechanism for PKM. So here's the PKM protein here. And so why is it that it's just the zeta gene and not any of the other genes of PKC that have PKM in the brain? Well, the answer is PKM protein, of course, comes from PKM zeta messenger RNA that gets transcribed from the PKC zeta gene. And the gene encodes the regulatory domain in exons here and the catalytic domain in exons here. Outside the brain, or the nervous system, the PKC zeta is made by transcribing this DNA beginning in the regulatory domain. But in the nervous system, and only in the nervous system, it turns out there's what they call an internal promoter that can begin here producing a messenger RNA that encodes only the catalytic domain of the PKC. So only zeta has that internal promoter of the 9 different genes of PKC. So that's why it's only PKM zeta that gets made as a PKM form. Now, this mechanism, in addition, explains how PKM zeta is increased during learning. And that's because this messenger RNA normally, under normal conditions in the cell, the resting conditions, does not produce the protein at all. It's actually a silent messenger RNA. And that's because it has a very long 5' end in which the ribosome can't get through. It's the structure of that 5' end prevents the protein from being made. But when the animal or the person learns something, then a special glutamate receptor called the NMDA receptor gets activated, which is not normally at rest during resting conditions of the cell, or normally inactive during resting conditions of the cell. And during learning, when there's a lot of activity of the synapses, the NMDA receptor opens up, allowing calcium to come in, and many, many other signaling molecules, those transient ones, are active, and they all are important for allowing the protein PKM-zeta to get made from its messenger RNA. Now, the action of this protein is, as I mentioned in my first slide, still the beautiful mechanism of being a constitutively active enzyme to perpetually enhance the synaptic strength by shuttling in extra glutamate receptors from outside the synapse into the synapse to double the number of the glutamate receptors. And it's even more beautiful as a mechanism than the simple model of the proteolytic cleavage of PKC to PKM, because the PKM-zeta messenger RNA making a protein can form a positive feedback loop such that the enzymatic activity of the protein now feeds back and enhances the translation of its own protein, forming a perpetual positive feedback loop that can maintain long-term memories for much longer than the lifetime of the protein itself, which currently we think is a couple of days. So, as I mentioned in the beginning of this chat, my father was right. And this is a fundamental discovery in how memories are formed and stored, but it has led to many other diseases, to an understanding of many other diseases. Even those that you wouldn't necessarily expect have to do with memory. Well, one of the first discoveries was that PKM-zeta was important, and perhaps the one most expected, in post-traumatic stress disorder, where there's an excessive amount of PKM-zeta made in different circuits of the brain. But surprisingly, there's also addiction. It's not the initial addiction that seems to be PKM-zeta is important for opiates or cocaine. It's the propensity to relapse. It's like even after one's kicked the habit, if you walk by a place where you used to get high, the temptation to relapse and be addicted again can be overwhelming for the individual. And that's thought to do with the memory, the positive, pleasurable memories that are in a different circuits of the brain. And it's thought that it's PKM-zeta in those circuits that's maintaining that propensity for relapse. Very recently, people have found that there are decreased levels of PKM-zeta in other areas of the brain during depression. Chronic neuropathic pain has been shown in many, many different labs to be also mediated by the persistence of PKM-zeta and other related PKC molecules, but in that perpetual fashion to maintain the pain state even after the inflammation of the initial injury has gone. And Alzheimer's disease, well, there's decreased PKM-zeta in neurons, but there's also, weirdly, increased PKM-zeta in supporting cells, glial cells. And in the protection of anesthesia and the recovery of function after stroke has also to do with increases or rearrangements of PKM-zeta. And so I think this is really how it works. One's curiosity leads to a fundamental discovery, and the more fundamental it is, the more implications it has for the rest of medicine. Thank you very much.